Hello and welcome everyone. I am taking a break from renal pathology since a viewer, rather student, has asked me to teach microscopic diagrams of the thyroid tumors. So I'll be taking two tumors which are important, which come in the exams. One is papillary carcinoma thyroid, other is follicular carcinoma of thyroid. So starting with papillary carcinoma thyroid in this lecture. So papillary carcinoma thyroid, it is the most common malignancy of thyroid, and the conventional papillary carcinoma thyroid, it can be solitary or it can be multifocal, and commonly occurs in the age group of 25 to 50 years, and radiation exposure in the head and neck region is an important risk factors. Grossly, it can be well circumscribed or it can be ill circumscribed, and on cut surface there can be areas of fibrosis, calcification, and importantly cystic change can be there. So this question is very commonly asked in the exams, and whenever we write the theory part, we should always draw a well labeled diagram. So I will try to simplify the diagram of papillary carcinoma thyroid. So to make a diagram of papillary carcinoma thyroid, we need to uh, know the different components that we will draw. So papillary carcinoma thyroid, as the name suggests, it is papillary tumor. So there the tumor cells, they are arranged in the form of papillae. So first important thing we need to know is how to draw the papillae. So the papillae, they are these projections which are finger-like projections, and they are having a fibrovascular core. So fibrovascular core means there will be fibroblast. Fibroblast, I already told you earlier that they have pointed ends, and in the center there is elongated nucleus. So basically they are spindle-shaped cells, and then there are blood vessels. Blood vessels, they are lined by the endothelial cells, which are again spindle shaped, and in the lumen of these vessels they will be red colored, these RBCs. Then they can be a formation of psammoma bodies within these fibrovascular cores. Psammoma bodies, they are a type of dystrophic calcification. I have taken them separately in the lecture of pathologic calcification, so you can refer to that. Now these papillae, they can be seen in longitudinal section or in transverse section depending on the way it is cut. So in longitudinal section they are seen like these finger-like projections which are decorated with these tumor cells which are cuboidal, and in transverse section, if it is cut transversely, it will be seen like this rounded structures again decorated by these cuboidal tumor cells on the outer side. Again the core will be fibrovascular in both. So this is how we will draw a papillae, and with the tumor cells which are lining the papillae, they will be cuboidal. Now next are the very important the nuclear features which are very characteristic of papillary carcinoma thyroid. So the reason why these nuclear features are seen is not fully understood, but maybe because of the RET-PTC gene rearrangements. So studies are still undergoing to understand the exact cause of these nuclear features. So what are the various nuclear features that we see in PCT? One is there is intranuclear inclusion. So these intranuclear inclusion, they are basically intracytoplasmic intranuclear inclusion. So there they are derived from the cytoplasm. So when there is a cell, the nucleus, it will show the grooving and it will enclose a part of cytoplasm within it. So since the cytoplasm is eosinophilic pink in color, so these inclusions, they are also pink in color. So these are the intranuclear inclusions, and their origin is cytoplasmic. So they appear round and they are pink in color. So these are intranuclear inclusion formed by the folding of nucleus, and they are enclosing a part of cytoplasm within it. The second, because of the folding of nucleus, they will be seen nuclear grooving. So nucleus has folded. So this fold is seen as a fine line which crosses the nucleus. So this is nuclear grooving. Again, very important feature of papillary carcinoma thyroid. Then the chromatin in the PCT tumor cells, it can be transcriptionally active. In that case, it appears like a open nuclei. So it shows powdery chromatin. So this is active chromatin, that is euchromatin. Then it can be transcriptionally inactive. It is called closed nuclei and consisting of the heterochromatin. And the third most characteristic is the orphan Ni nucleus, where the chromatin is seen in the margin. So the chromatin is arranged in the margin, and the center part of the nucleus, it is clear. It shows clearing. So optically clear nucleus is seen, and its name is famously called orphan Ni nucleus. So orphan Ni, it was a comic strip which used to come in America, and the main protagonist of this comics is this Annie, and her eyes used to look like clear. So again, resembling this, because of the resemblance to this appearance, these nuclei, they are called orphan Annie I nucleus. So these are the types of nucleus that we will draw in the lining cells of these papillae. Okay. Then lastly, PCT can show SEMO bodies, though they are not diagnostic feature, but they can be seen in this papillary carcinoma thyroid, and they are seen in purplish concentric laminations. So they are seen like this concentric laminations, and SEMO bodies are important feature of papillary carcinoma thyroid. So these are the features which you should know how to draw, and when you know these, then you can easily draw the diagram of papillary carcinoma thyroid. So let's now switch to the making of this diagram. So let's start. So you make a circle. Then first of all, we make the papillae. The papillae we will make first in the transverse section, longitudinal section, both. Now here we are making the blood vessels which are present inside the papillae. Blood vessels, they are lined by the endothelial cells. They have elongated nucleus. Spindle shaped cells they are. Then we can make RBCs in the lumen of the endothelial cells. Then again in the transverse section of the papilla, again we draw the blood vessels because it is fibrovascular core. We also make the fibroblast. These fibroblasts, they are spindle shaped cells having pointed ends, and the nuclei is again elongated spindle shaped. So this is how we make the core of these fibrovascular cores. Now these papillae, we will decorate them, or we will make the lining of these papillae by the tumor cells, which are cuboidal. Cuboidal cells means length and width are the same. So they are squarish, they are like a square. So these cuboidal cells, we can also draw focal overcrowding of these cells, which is seen in papillary carcinoma thyroid. Now lastly, the nuclear feature. Now you make the nuclei, different kind of nuclei, as I explained earlier. Some nuclei you can show the orphan Ni with central optically clear nucleus. Some in some nucleus you can show the intranuclear inclusion. Then in some you can make the open phase nuclei or closed phase nuclei. So first you make the nucleus, then in some you can make the grooving like this. Like this you make the intranuclear grooving. In some you make this open chromatin. Now again you make the nucleus in all these tumor cells, and different types of nucleus you have to make. Now this transcriptionally active nucleus, then clear nucleus, then this again intranuclear eosinophilic inclusions again you can make, and then you make the labeling. So these are the tumor cells, cuboidal, lining the papillae. So tumor cells lining the papillae in the longitudinal section and transverse section. Then you label the nuclear features like nuclear clearing, known as orphan Ni nucleus. Then intranuclear cytoplasmic inclusion, which you have to draw pink, and nuclear grooving, and lastly the fibrovascular core. So this is how you make the diagram of papillary carcinoma thyroid. And one more thing that you have to make is the Samoma bodies. So you make the Samoma bodies in the fibrovascular core. So you have to make the laminated way. So these are the concentric laminations. So these are the Samoma bodies, and you also label it. So this is the complete diagram of papillary carcinoma thyroid. Hope you are able to understand it, and any questions are most welcome in the comment section. Thank you very much.